Hello! Welcome to Microbiology Journal Club. I'm Faz Alam, and this is my colleague Danny Chan. We haven't thought of a good intro for us yet, but it's a progress thing. Uh,
Yeah, this we meet every week to talk about microbiology stuff. Our backgrounds are both we did graduate school in microbiology and infectious disease. I studied Staph aureus on skin culture.
yeah, I studied flesh-eating bacteria, making it glow in the dark. Um,
And
so,
during this time, we came together to try to talk about news, like to keep our knowledge up to date. And we thought it'd be a nice format to catch other people up as well. This episode is our off week, so called, where we look at the news headlines, and then from this we'll choose an article for the next week where we'll dive in figure by figure and talk about some of those details.
yes, indeed. And I feel that this week is going to be a week of the COVID-19 Vaccine Olympics
Yeah,
because
yes. Wait, do you have that
we've
overlay still? I
I,
forgot to
I do
ask
have
her.
that overlay still. I've snapped it up right now. So
That's wonderful.
yeah. Uh, so yeah, it's a basic thing it's a segment we do to talk about the latest COVID vaccines and how well they're doing. And well, this past 2 weeks we've had a lot of vaccine news coming up,
Lots
and so I
which
guess
gets people
we'll
very excited, right? I think in a lot of earlier episodes we said like everyone wants to know like when can we stop doing this stuff? When can we stop social distancing? When can we stop wearing masks? Well, don't get your hopes up because vaccines are like not exactly that. I don't know if we've talked about on this stream. I don't think we have, but have you seen the Swiss cheese model of like preventing COVID-19 spread?
yes, I do. I know. Yeah.
Yeah,
My
the
old
idea is that
super
nothing's perfect, so like everything has limitations, and we're using them all in combination to deal with this outbreak.
Yeah, I guess I should do a shout out to my old supervisor who has done a lot of Susie Wiles, who's done a lot of work to promote that Swiss cheese model. But yeah, the basic idea is like you need like not just one filter, you need multiple filters in order to make sure that you're safe. So even if the vaccine is out there, it doesn't work 100% of the time. So you'd still want to have a face mask, or you still want to do social distancing until the number of cases in your area are low enough that
Absolutely.
you can feel safe.
Yep.
Uh, so, but, I mean, if you're in New Zealand, you don't have to worry. You can just be smug.
Yeah, you're smug. Well, I'm sure they're taking it seriously. I mean, like I think New Zealand is the way it is because everyone has taken it seriously, right? The
Yeah,
government and the people. So, yeah, it really requires like quite a complex alignment of social forces to have things go well.
yeah, you have to have a bunch of people do the right thing, and that's apparently a lot more difficult than you think. But
Yeah,
enough.
let's dive
So
into those headlines.
let's dive into the Moderna vaccine. So last time we did this show, we talked about the Pfizer vaccine. They just released their results. I know Sputnik released a couple of results as well, uh, so we talked a lot about the Pfizer vaccine, and the day after, Moderna released their results. That we didn't get to talk about that properly.
Yes.
So they released their.
A proper like media dogpile of interim efficacy endpoints.
Oh yeah, I can literally imagine someone at Moderna going, "Oh, biotech. Oh, shit. We need
Yeah.
to release something."
I mean, from my experience, right? Like currently, I fact check pharmaceutical ads in
Mm,
an advertising agency. My suspicion is that they sort of had their right. They have like a package that's
mm,
ready, right? And they're like, we want to wait, right? To if they were going to be the first ones, they would they would release it and it would have the exact story that they wanted to tell. They were waiting for like some finishing touches, but
mm.
because Pfizer released, right, they were like, all right, we can't wait for the finishing touches. Just put it together and get it out there.
Yeah. So yeah, the Moderna vaccine, they said that they got an interim analysis based on 95 cases, of which 90 cases were in the placebo group and 5 cases were in the vaccine group. So that looks pretty good. Vaccine efficacy of about 94.5. And so they basically like
Yeah, they're
so.
saying the same thing. And it's the same thing as we said 2 weeks ago as
Mm,
well, right? Like this is a case where we're waiting to see cases, right? Like the statistics that people are looking for to prove vaccine efficacy, right, is that over this period of time, eventually people will be getting positive SARS-CoV-2 symptomatic or whatever, right? And they're going to be in one of two groups, either the vaccine group or the control group.
Mm.
And after they accumulate a certain amount of cases, right, they'll see how is the distribution spread between those two groups.
Yeah.
And that's what you just told us right now, right? As of this time point, November 16th, right? 5 of them were in the placebo, and then however many in the.
Yeah. So in order to like get the statistical significance, they want to collect 150 total number of like cases, and then work out the
Yep.
division. But like now that we're like we've got 90 of those cases done already, even if all the other cases come back within the vaccine group, that'd still be 60%. So that would still demonstrate some degree of efficacy. So
Right, right. Oh yeah, earlier, yeah. I said 5. I meant 5 in the vaccine group, right? That's what you said.
yeah, sorry, yeah, 5 in the vaccine group.
That was my
Um,
bad.
And they also like added some more information, like a secondary endpoint analyzing severe cases. Now, I think severe cases have to have like a bit of because how you define severe cases does change depending on who you talk to and which. So I think but it still looks quite optimistic because apparently the 11 severe cases were all in the placebo group. Uh, and they also take great pains to acknowledge that the cases included people in the age 65 and older age group, and that people were from more diverse communities.
Mhm.
So
Yeah, I was just gonna say yeah, I just asked you earlier, right before we started this stream for the Moderna protocols, because I was like, oh, I hadn't seen them, and I have the severe definition pulled up right in front of me. It says respiratory rate greater than 30 per minute, heartbeat 30 per minute, like low SpO2,
mm,
ARDS. They have to have ARDS.
yeah,
They have to be on the ECMO. Um, sorry, ARDS as defined by the need for these severe interventions.
so
They
that's
also
acute.
have to have severe acute renal or hepatic neurologic dysfunction. Um, yes,
So
or in an ICU.
yeah, so that's all really bad. So ARDS, for people who don't know, is acute respiratory distress syndrome, uh, and that's really bad. You don't want it.
Yeah,
In
I mean
fact,
that
all
was the primary cause of mortality, right?
yeah,
With our first wave, probably still is the primary cause of mortality, right? Is the mechanism of injury.
yeah, and so I think that the fact that these results are very positive news, but then Moderna followed up with another like press release not shortly after about their vaccine has a longer shelf life that uh,
Hmm.
so this is a real dunk on BioNTech's vaccine because
Yes.
I think the thing that's been hitting the news a lot is how BioNTech needs things to be at minus like 70 degrees,
Hmm.
and there needs to be a lot of infrastructure put in place because you can't just put it into any old refrigerator.
Hmm.
Um, Whereas Moderna, they've come out with basically saying that they've run tests and they expect that their vaccine candidate is now expected to be stable at refrigerator temperatures. So
That's crazy.
yeah, so
Especially when you
two
think about, mRNA, right? Like this is this molecule that usually lives inside of cells, so we don't think of that as a particularly stable thing.
yeah.
Um, but yeah, I guess advances in chemistry can probably bring us there.
Oh yeah, I went into a real. Well, we'll get to that because I went into a real like mind hole trying to find out what the why the because BioNTech and like on paper both these vaccine candidates should be somewhat similar,
Hmm.
but
Yeah, they're
but there
both
are
RNA, lipid, um, lipid nanoparticle form delivery. They both make the spike protein.
yeah, exactly. Yeah, one has to be kept in like a minus 80 refrigerator, really, or like with dry ice on alcohol, just like really really cold, or the other one can be kept in like either your freezer for about 6 months. They predict. I mean, it's hard to predict these times because this outbreak hasn't been like longer than that much longer than 6 months. So, I
Yeah.
mean, they do like have to revise these every so often.
Hmm.
And the other one, and you can actually leave it in your refrigerator for about 30 days apparently. So why is that? So this sent me down into a hole where I went to the Moderna website and looked through their patents to try and figure out what is up with their vaccine candidate. And
Yeah, it's
so,
cool that they provide their patents. I guess they definitely want to let people know that do not, you cannot copy us in these particular ways. This is like, yeah, for their, it makes sense like from the business
yeah,
perspective. And there's always this, I don't know if you've ever heard about this about patents. People say that they're very different, right? Like when you learn things, it's hard to learn things from patents because they're written to be the most general as possible, right?
mm,
They want to cover as much IP, right? So you don't get always a sense of what is very specific about them. Um, Sometimes you just get like a whole array of different processes that might come together to create the product.
yeah,
And it's true, like scrolling through these, there are so many compounds shown.
oh yeah, and that's the interesting thing about this patent because they go a lot into their formulations, specifically on like the lipid nanoparticles. So
Hmm.
so Moderna is primarily like an RNA based company. They're very into to RNA therapies, not just for vaccines but for other treatments. So this is a kind of a question
this
they've
is
been
this is what the company is built around, right? This
yeah
particular platform.
yeah
And so, like, if people if older viewers recall some of our past episodes, we talk when we talk about vaccines, especially these ones that are DNA based or RNA based, you have to get that into a cell in some way, and the technology that Moderna is based around is this lipid nanoparticle technology.
yeah. And BioNTech also have that lipid nanoparticle technology, uh, but they probably do something slightly different. I mean, I couldn't
Well, I
find
mean,
any.
I was going to say, if the patents exist here for Moderna's formulation, then BioNTech is not these molecules,
Yeah,
right?
it
Like,
cut. That's that is the rub. They
threat of being sued, their
yeah,
molecules are not these molecules.
and that's again another issue for BioNTech because Moderna, since they've been doing it for so long, it means that they can pip to the other companies to the punch on a lot of things.
Yeah, I'm not
Um,
sure. Is Moderna newer or is a older company than BioNTech?
I feel like Moderna's been around for maybe a decade. Uh, working on very because I do remember they were part of they got involved with the Ebola outbreak in 2014. So
Interesting. Interesting. Yeah, so
uh,
they've been. This is sort of like more of their focus, and actually, I guess I should research more into the company. But like BioNTech also, this is an acquisition, right, by Pfizer.
yeah,
So like, there's also it's interesting to think about the different business like attributes, right, of the two vaccines in that way. Okay. Anyways,
yeah. So the thing that I came across was like the as a function of the lipid nanoparticle, they talk a lot about these aminolipids. So I went into the website further and I found this lovely presentation where they talk about all the about how they structure their lipid molecules, and they pull out a bunch of structures about well they modify I mean both companies modify their RNAs in two ways. They modify the actual chemical bases. So BioNTech uses uridine instead of uracil and does a few other modifications. Uh, Moderna like have a whole bunch of different chemicals that they work with as like replacements for mRNA.
mhm.
But so
Yeah,
that's a
so like normally RNA is made up of the four, right,
yeah
ribonucleoside bases.
yeah adenine sorry I forget so uracil
Adenine, thymine, guanine, uracil, yeah, but
yeah
instead of with the ribose sugars instead of the deoxyribose sugars.
yeah so I
But
couldn't
there's further changes,
yeah
right, that I've made. Yeah. I remember when
so
we covered the BioNTech, we read some of those changes on that side. Moderna
yeah
also does it. Do you think
yeah
that that could be the stability, the key to their stability?
I think that's key to their stability inside of cells because I
Right.
think a
That
lot
would be
of
my thought as well. Yeah.
so I mean feel like it might have an effect on other versions of stability. I mean I've had people from sources say that, but
Mhm.
my sense is that it's the lipid nanoparticles that because like
Yeah.
RNAs are big noodly things that get wrapped up, and part of the things that makes them unstable is because all of those bases are exposed to the environment, and you can
Mhm.
also get kinks, and it can. Whereas if you enclose it in a nanoparticle by then you can actually like not only protect it, but you can also like force it into a specific conformation that prevents it from wobbling around too much.
Sure, sure. Yeah, I was also thinking it's possible that some of the base changes could like help the binding between the lipid nanoparticle and the RNA molecule.
yeah, I feel like they definitely think about that when they make their formulations in terms of because they use different lipid nanoparticles. So I think that they might actually try and think about how they're going to the kind of structures they're going to create with them by modulating how much of
Sure,
each.
absolutely.
Because so one end of the nanoparticle has this ability to do hydrogen bonds on and bond directly to the bases. But also I
Hmm.
noticed they had this thing called a squaramide ionizable lipid, which can do another form of bond. So I've put
Sorry, what was that term? Square my
squaramide because it's got a
square.
square structure on it. I don't know whether they use this in the vaccine or not. I'm
I
just this
see.
is they
But
talk
that's
about
just from
this.
diving into patents. You saw like they have a very weird formulation of lipid molecule.
Yeah. So this one is a bit. So I'm posted like some images up on the screen of this molecule that they talk about
Yeah.
in this open day they had with their investors. Um,
Hmm.
and they've made it fully public available, so you can actually like listen to it yourself. So I didn't get to listen to the actual like uh, blurb that comes along with this, so I might be a bit wrong. I'm just going by the diagrams, but essentially they say it can like bind in like by hydrogen bonds and also by pi stacking. So
Ah.
when you got pi
Ah,
orbitals,
interesting.
yeah,
Yeah, that's yeah, like a four-member carbon ring could intercalate right into the RNA potentially. Huh.
yeah, and then hold it in a specific conformation until such time as it needs to be opened up. So uh,
Yeah.
I think they think quite deeply about the structures that they work with, and I'm sure
Sure.
that BioNTech have the same thing. But
That brings, I mean, of course, like all of this stuff has been validated, right? Also before this, right? They were
hmm,
working on vaccines like this, but yeah, things that intercalate, I'm like, oh wow, that's interesting from the safety consideration, right? Because like having those types of molecules around the cell, I would wonder what the interactions would be. Um,
yeah,
but of course, having them already at this stage means that they've passed at least these preliminary tests.
yeah. So
But I think maybe an interesting point that people might not think about is that that means that the adverse effects profile, right? Like
hmm,
when we say, oh, they're essentially the same thing. They're lipids, mRNA, right? Lipids and mRNA that we're injecting, right? That encode the spike protein. All of that sounds the same, but these small differences, right, really might mean that there are different adverse effects of
yeah.
these vaccines.
I mean, we do know something about the formulations, but we don't. I mean, there's all sorts of other things that they talk about, like the size of the nanoparticles, the amount of DNA that's in them. So
Mm,
Moderna has about 100 micrograms in per dose, whereas BioNTech has 30 micrograms. But
mm,
BioNTech talk about their self-amplifying, so their mRNAs can self-amplify inside cells, so maybe they don't need as much.
sure.
Um, and so, I mean, a lot of this is just like speculation because very little of this is publicly available, and a lot of it is proprietary information. So I'm just trying to deduce based on like stuff that has
Yeah.
been published previously.
Yeah.
Um,
But I mean, again, we know that there must be differences. Like, if
hmm,
these claims, if we're to believe the claims, right, of the difference in shelf life, then it's driven by some sort of chemical difference. And so
yeah,
this is just like our stab at reading,
yeah,
diving what they were saying. Yeah.
and and to me, it seems like reasonable that it is the way that the lipids stack together and the way that it kind of almost binds specifically for these RNAs.
Sure.
And
Increased binding between the right, increased binding between the lipid nanoparticle and the mRNA would mean more energy needed to break it apart, right?
yep,
And so more energy would mean that it have to be at a hotter temperature, which means it could sustain, right, some small excursions into a higher temperature.
well, yeah. I mean, as long as it can hold this conformation at around refrigerator temperature, but then lose it when you get into 37 degrees, then it's okay.
Well, it's not 37, right? They actually have to keep their shape like while they're like they have to find the cell first, right? Because it's
Yeah,
the
that's true.
lipids, it's the lipid outside that's mediating the transition of the right from the extracellular into the intracellular. So
Yeah.
so I mean that's like at some level when we think BioNTech is held at -80, right? Like that's it's gonna experience 37 degrees when it's in the body, right? And it's good enough at least, right, under
Mm.
those conditions to get the mRNA into the
Yeah.
cells, right? So, it could also be that BioNTech just never performed those types of tests to be able
Yeah,
to make that claim,
that
right?
is
And they've been working from like the laboratory conditions where it's like, okay, -80s are everywhere in the laboratory, and in their rush to bring it to market, they sort of didn't realize that this is a good marketing axis to talk about.
yeah. I think that is a brilliant point you brought up there, because it could just be that this is something that Moderna thought a lot more about, and so they have already in place. With BioNTech, they could potentially have a vaccine that is just as good. They just haven't had the chance to do the test because, I mean, how can you predict 1 year stability when it's not even been a year since the
Absolutely.
COVID has
Yeah, and
been.
I think this is where this idea of like Moderna as a platform company really like comes to mind, right? They've been working on mRNA in lipid nanoparticles for a variety of different applications. They've had years, right, to
Mm,
sort of figure out like the best combinations. Whether or not those combinations are like better than what another one could, they can show it, but I guess we don't know
yeah,
technically.
they've got everything in place to be like, oh yeah, this is our strength as BioNTech. They could have a genuinely much better vaccine, but we wouldn't know until. I mean, with all this, we only have press releases to go off of, so we don't know the full story yet. There,
Absolutely.
there could be,
Absolutely. Yeah.
yeah,
Like the scientists at Pfizer BioNTech, they could be so convinced, right, of like
mm,
the values of their of their vaccine, but without that data to show us, right? Like, well, they just got to go forward with what they got.
yeah. Uh, so I think we've. I think it might be time to talk about AstraZeneca slash Oxford.
Sure. Yeah, yeah, yeah. Let's
Yeah.
switch gears and talk about that. So
So
this is we've actually talked about this vaccine before, I think.
yes, the Chadox vaccine.
The Chadox vaccine,
I
yes.
think we talked about
Renamed
in.
because I guess that Chad. I bet that Chad thing didn't go down
Yeah,
well.
the
um,
Chad vaccine.
yeah, and again, maybe that from both the perspective of the internet's parlance, but also the fact that like it also denotes like the chimpanzee adenovirus, and that
Yes.
because when I talked about that to another group recently, they were like, "Oh, I heard that the chimpanzee adenovirus is isolated from stool." I'm like, "Oh, okay, but what is that? What relevance does that have to the vaccine? Like that's many passages ago."
Yeah. Oh
um,
man. Like.
but yeah, so if you remember from our previous episode, the quick recap, right, is adenoviruses are DNA-based viruses, right? They deliver DNA into our cells. That the reason why they went for a chimpanzee version is because we have some natural immunity to them, and so they were hoping that by going with the chimpanzee version, we'll have less of an immune response towards the vector, and yeah, more I guess immune response towards the thing that they're delivering.
Yeah, this has been a real problem for other adenovirus. So the CanSino vaccine, a lot of people had already got human adenovirus before that, so they weren't going to get any. So they
Yeah,
they didn't react to the
basically,
vaccine.
you wouldn't even get to the stage where your body was making enough of the spike protein to mount an immune response. And so that's what this technology is hoping to sidestep. So very different from BioNTech and Moderna, right? We're talking
Hmm.
about viral vector. We're talking about DNA-based construct. Not, and I think an important thing to also say is these are non-replicating. At least in Chadox, it's a non-replicating
Mm.
viral particle. The only reason this virus coat is around is basically essentially serving the same purpose as that lipid nanoparticle. It's facilitating the transfer of nucleic acid into the cell.
Yeah, uh, and so like there's been a lot of hype about like the Chadox about the AstraZeneca vaccine in the UK because it's tied to Oxford, which is a very British place, and but
Institution.
yeah, the most British of places in Britain. Um, um, but yeah, so they did a dosing regimen of like. So actually, this is weird, a very weird press release that they sent out because they sent out the results of not one but two different studies, and then reported them as if they're one study.
Mm,
So basically overall, the headline amount was that okay, it was about 70% efficacy.
hmm.
But that was actually split between two groups. One that had a vaccine efficacy of 90%, another group that had a vaccine effect of about 62%.
Yes, they
So
average them together.
yeah, that's a pretty big dip in numbers. So that
So that could be because of enrollment, right? Like,
mm,
you know, you try to enroll like really balanced groups usually, but
Hmm.
that's not always the case. Or did they also specifically enroll different groups for these two
I
efficacy
I
endpoints?
mean I will get to that later, but so um, 'cause they didn't release that information initially.
I
They
see.
basically said what they said was that one dosing regime was 2,741, so that was the one with the 90% efficacy, and then the other group was much larger, had 8,895, which showed 62% efficacy.
Hmm.
Uh, and the thing that they talk about that as the reason for this was that it was a one of those, as a story, it's like, oh look, one of those happy scientific accidents where someone did a dosing error in one of the studies. So,
Ooh.
yeah, so the one with the really like high 90% had the patients were given half the vaccine dose because the people giving the vaccine made a boo boo when they're doing the calculations. Made a
Yikes.
Yeah, and turns out that that turned into a much higher 90% efficacy, which
Hmm.
is interesting. And so now all the other studies want to do the half dosing regimen as well
Sure.
going forward.
Sure.
Um,
Yeah. When it
which
comes to this specific vaccine, right?
Yeah, when it comes to this
Yeah.
specific vaccine, and the one of the theories that was posited was because because adenoviruses are quite immunogenic. So the
Hmm.
thought was having an initial immune response that was really immunogenic means that there was lots of antibodies against the vaccine, so that when the booster came, it wasn't as effective.
Sure. Sure.
Which
That's
was
cool. Very
Yeah,
interesting.
but I think that then more data came out, uh, and it turned out that actually the key issue with this so is that there was the first group had an age cap of 55,
Hmm.
so no one over the age of 55 was included in that earlier group.
Mm. Yeah,
And
that I mean that's sort of where I would that's initially where my mind went when you said
yeah,
two groups with two different ones. Because I mean like it just happens, right? Like that's clinical trials are very complicated to do, and
yeah,
there's certain enrollment criteria that people place both for the safety of the participants
mm,
as well as right to demonstrate good results. Um, and so it might have been that when that was first made, they were like, "Oh, we don't want to test really old people because we're afraid of like giving them this immune response, right?" And then maybe the next one was made, and you're like, "Ah, whatever, whatever battle happened, like they were like, go ahead, do it with olders because also there's
well,
pressure, right? We want to know that it works in older people because those are the people that are most likely to convert to severe disease."
I think that in practice they just tended to like run these studies simultaneously, and certain like groups get results back. So it might just be that they paired off this younger group, but they have an older group on the side, but they haven't got any results from them, or they're not releasing any results from them.
Sure, absolutely.
But either way, this is not a great way to present their data because with the other ones they are literally just presenting with one single trial protocol and one like kind of group that's been done across
Right,
multiple hospitals. Whereas with
right.
this study, it tends to be that they're doing two different groups, two different study trials that have been run slightly differently, and then
Hmm.
reporting them as the same. You're basically getting an apples and oranges situation.
Yeah,
Uh,
yeah. But you could also think of them as two. I actually I like it better to think of it as two. I actually like it better because
mm,
two trials, right, is two different conditions, two different protocols, right, in which that we see results. Of course, the numbers are more meaningless, right,
mm,
because we don't really know which number to go with. But I like the independent verification part of this, right, because right, like
yeah,
I think that actually makes for strong data, right? It's as if it was
yeah.
a replicated study.
I think I agree with you in that, and
Hmm.
I think that if we had these as like published studies, they'd be much stronger. But
Absolutely.
I think
Wait,
this
don't we
also
have one as a published study at least?
we have one published study that's a phase 3 trial that does well, a phase 2 trial that it doesn't talk about the actual efficacy data of the vaccine. It's just mostly
Mm,
safety.
hmm.
Um, so
Right. It's the safety reports from right from the same cohort that is being studied in this way.
yeah, so this is
Yeah.
from the CoV-002 vaccine. I couldn't really pick out from my own thing which which studies these cohorts reported in the press release came from. Um,
Mm,
but yeah, that's the issue with getting things from press releases, because there could easily be stuff just as bad in the BioNTech and Pfizer stuff, but because we don't have all the data available,
Yeah,
that
for
and
sure.
yeah, so it
Yeah, like it could be that. Yeah, I mean, it's interesting, right? Because yeah, press releases don't have the same level of rigor, right?
mm,
So in the press release of BioNTech and Moderna, not to say that they did it, we assume at face value they did what they said they did, but
yeah
they could have merged two separate studies into a single thing and just chosen not to report like that they did so, and we
yeah
may only see that once something gets published.
exactly. So, but
The other thing that maybe we said 2 weeks ago as well is that there's a lot of we have to have trust in our regulatory bodies
mm,
at this moment, right? Because it's the regulatory bodies that are giving the green lights for bringing these
mm,
vaccines out, and the companies have to prove to them first and foremost,
yeah
right, that their vaccine is safe. So it takes a while for it to trickle down to us, the general public, which is unfortunate because
mm,
the general public is full of inquisitive minds. We could help, right?
yeah
Make some decisions, or at least like help people understand why the decisions are being made. Um, but yeah, anyways, we have what we have.
yeah. But I mean, either way, the way this has been like the way that this press release and every all this information has been leaking out, it does not reflected well on AstraZeneca and well, actually this trial. So that's the main issue. I feel
Yeah,
like
it seems like the to me the takeaway from the news is kind of like the trials were done a little bit maybe in a rush, maybe kind of sloppily, right? Um, and so yeah,
I mean,
we
I
have
feel like it's more like they had to release something because of the press releases because they wanted to compete with the other thing, and these trials aren't finished yet, and they suddenly decide to pick out the best results for the press release without thinking about the scientific implications. Because, I
yeah,
mean, this vaccine has already been delayed like a month due to like a safety scare, so
yes.
they're already operating like on the back foot. So I feel like their press release department came under a lot of pressure very quickly and ended up coming up with something that sounded good at the time, but ended up reflecting badly on them.
Agreed. And I'll also say this though, that like um, the fact that they are feeling like that they had to the fact that they paused for those safety concerns and were
Mm,
put on a back foot, that's also a bias in I mean, they did the
mm.
right thing, right?
Yeah,
But that's also a bias within the regulatory community, right? Because we've seen adverse events from these types of vaccines before. So
mm,
people are looking very closely, right, for those adverse events because
yeah,
they know that they can happen. But in lipid nanoparticle formulate delivery, there's not as much known. So
yeah,
like people also aren't as attuned to like little things. Be like, well, we gotta wait, right? Like that would never happen because yeah,
yeah,
the technology is just a lot newer.
yeah. I mean, lipid nanoparticles don't have a body count like adenovirus has,
Right.
so I think that's
Right.
the main thing that focuses people. Uh, but yeah, it's difficult, and we won't know that. And if there is a body count, we will find out very soon. I feel,
Mhm,
but
mhm, mhm.
yeah,
Um, I will, but remember, I think that the other important thing is that when you say body count from adenovirus, that
yeah,
was during clinical. Yeah, that's one,
yeah.
and
One
during a
one one
clinical
guy.
trial.
That was
Yeah,
during a
right.
clinical trial.
So like, so in terms of like the RNA vaccines, lipid nanoparticles, they've been through those clinical trials. They did not see fatalities, right?
Yeah,
So that's not the type of side effects that you would be thinking about there.
yeah,
And again, a reason why that they didn't experience the same pause, right? But just that one is enough to make pause in this version. But
yeah,
whether or not, again, that's a certain type of regulatory bias. When you know the dangers out there, you are more cautious for it. When
yeah,
you don't know the dangers out there, you do as you normally do.
yeah,
I
and
think I always think the best thing to look at in these cases, the best way to assess risk in these cases is stuff like Vioxx, right?
mm,
Stuff that like got to market with like
yeah,
not bad efficacy, but when it was tried in the larger population, did have like some strange long-term side effects that people
mm,
weren't aware of.
yeah, and there is that potential for because when you're dealing with any genetic vaccine, you can if you get paranoid enough, you can concoct any way for it to cause some problems in the future.
Yeah, yeah, absolutely. But I'm hoping that with like this the emphasis on the non-replicating kind of standpoint
Mm,
is hopeful, right?
yeah,
Again, it's like I feel like a lot of this is scientific, this is decision making, when decision making meets science, right?
Mm.
Because as scientists, like, we want to be super conservative in the interpretation of the data, right? We always want to do more tests, more data,
Yeah.
right? But then you hit the reality and people have to make decisions. It gets really messy because there's always going to be trade-offs, right? And what I care about as a reader of science is I care about like orders of magnitude, right?
Mm.
Change, right? And I'm when it comes to decision making, I kind of see that, yeah, there's lots of weird regulatory biases that exist, and like we should be very acutely aware of like when we're talking about issues that are from those kind of concerns, like trying to be conservative, versus when we're talking about concerns from like an order of magnitude danger, right? Where it's like, oh, the danger is there, and for the most part, like I think we have to try these vaccines. I think that these are very important tools
Yeah.
in our toolbox against this infection. And so, yeah.
Yeah. I mean, I think that you're also thinking about like relative danger as well, which is also an important like concern because every minute you delay, people are dying. You need to kind of price that into what your decision making, and that makes things a lot more challenging to think about.
Yes, yes,
Um,
yes.
but
And
yeah.
in that way, I sort of argue that we should have trust in our regulatory agencies, right? Because like
Mm.
the regulatory agencies are the ones that are setting these endpoints, right? They are
Yeah.
saying that these are the bars you have to pass. They're the independent verification on the company's documents, right?
Yeah.
And I would love to be that part of that system and also independently verify, but actually the whole speed of things is not making it conducive to do so, right? The
Yeah.
scientific community is not being leaned on the same way as it normally would be, right?
Yeah.
To validate these things. Investors are sort of like dealing directly with the data of the company, and not we're not seeing like that normal cycle of like, oh, then we see our phase 3, then it went into regulatory, and then they decided. So so yeah.
Yeah.
I'm excited about the vaccines. Maybe
We.
that's how I'd like to end this section, right? I'm excited. I think that there
Yeah.
is a lot of benefit that they're shown to provide, um, but I also think that there are many things that they are not showing us with this data. And I said I'd be on my soapbox about this for a while. Is that like they are testing for symptomatic? We looked at the Moderna protocol and some of the other protocols maybe 2 weeks ago. They're looking at symptomatic disease, right? That's one of the ways in which they mark, right, for their primary efficacy
Mm.
endpoint, right? That 90% of people who don't get symptomatic disease, and so think about the people who can't take the vaccine. Think about the
Mm.
immunocompromised. Think about the people that weren't included in the study who are too old, right? Where the
Mm.
vaccine may not be as effective. Those people will not ever get the vaccine, right? Or maybe they'll get a vaccine, but it's like much further away for them
Mm.
than it is for everybody else. If these vaccines do not address asymptomatic spread, right, and only symptomatic disease, those people are still vulnerable,
Yeah.
and our adherence to social distancing practices is still extremely important because we could be transmitting the disease to those people by some crazy epidemiologic chain, right? And so, like, I'm excited for this vaccine. I'm gonna take the vaccine when it comes out, right? But I want to make sure people know, right, that just because you have the vaccine, it doesn't mean your life went back to normal, right? Because there are still like greater forces at play here, in terms of the spread of the disease and those who are susceptible to dying from it.
Yeah, I think that's a great point. I don't think I can add anything to that. I think that's
Yeah,
perfect. So,
yeah. I've been like building up this rant. I'll probably like deploy it several times over the course of the next year.
yeah, no, that's fine. But yeah, so in all, like COVID vaccine Olympics, it's not decided yet, but it's getting there. There's a lot to be
Oh yeah,
optimistic about.
yeah, yeah. We
Yeah,
definitely. I think the Olympics is made up of different categories. Maybe the COVID
yes,
vaccine Olympics should have different categories, right? I would say definitely golds have been won in the
yeah,
vaccine Olympics, right? But as to like the final country of right most golds, we have no idea yet, and
yeah.
there's a
We
lot more time to find out.
ah. Yeah, so now we're going to move to the second section. We talk about papers that have caught our interest, interesting scientific papers.
Yes.
Um, so the first one that caught my eye was okay. Well, there's a new like Chaddock's paper, but But I think the another one
I
that's
do. We should talk about that one. I
yeah,
think that that's. I mean, we haven't typically talked about. I think I got tired. Maybe we got tired of
yeah,
diving into clinical trial papers because we don't get to talk about those fun things that we know about in science, right?
yeah,
Mechanism and pathogenesis. um, but again, this is the one that I had mentioned, and you said you couldn't map it to the press releases, so you're not really
yeah.
sure where this cohort comes from.
I'm
But
yeah,
likely it is one of the cohorts, or a piece of the cohort at
yeah.
least.
I get the impression that maybe it's like this because this has like an younger cohort and an older cohort, so it could be that this is like in the press release they reported half this study, and then the other half is coming later. Um,
Yes, yes.
so
Yeah,
it's
so this could be that half. And again, here it seems like they're focusing on the easy stuff that they can show, right? Like
mm,
the safety and the immune response, right? So of course we know that these vaccines make antibodies when we give it to people,
mm,
but the big question is, are those antibodies effective? That's what we're getting at with the phase 3 data and the whole like waiting for cases to roll in.
yeah,
Cool. But actually, I mean, it's an interesting paper from the standpoint of I love like it's nice to see this is like the most phase 3 of the published data out there,
yeah.
right?
I mean, yeah, I'd agree with that. I mean,
Uh-huh.
yeah, I think it definitely is. I mean, I think this is the only one that has like a phase 2/3. So there must.
Mhm.
So I'm trying to figure out what the /3 part of it is. Because
To
we've
me, I
got
think I believe the /3 probably just indicates that it comes from a cohort that will end up in the 3 analysis. So
yeah,
like basically like these antibody responses, they're going to end up being correlated against the efficacy that we see in a phase 3 study.
yeah. Uh, I
But
definitely
but I
yeah,
don't know. I don't know for sure. I think it takes a bit of digging.
yeah. Um, so there's that.
Okay. So that is one. And then let's see what's next. We have complete mapping of mutations to the SARS-CoV-2 spike receptor bind domain that escape antibody recognition.
Yeah.
Okay.
So this kind of like cottons onto some other things we've been talking about. So we talked a lot about like the variants of SARS-CoV-2. We talked about the D614G mutation, and
Mm-hmm.
and a bit about like antibody escape.
Mm-hmm.
I mean, last week we talked about the ACE2 variant
Yeah,
that was used.
this is the next paper in the sequence um, that we talked about last week, right? Last week we were talking about escape mutants, I believe. Let me
Yeah,
just double check that because I see a similar author, this Tyler Starr fellow. He was first author of the paper that I referenced last week. Um, so
yeah,
let's just see when that was published. Deep mutational, yeah. So this is the paper that we're discussing now is published in October. The paper that we discussed last week in the introduction published in September. So
right.
this is very much an extension of the same work they were doing because I see the same techniques at the beginning too. They're using
Mm,
yeast expressing RBD domains, right? Doing like deep saturation mutagenesis, but now they're taking it against surveil for variants that alter antigenicity. Um, so
yeah.
inside of people, they're actually looking at convalescent serum.
So yeah, based on the visual abstract, I think so, but I'm not sure. I didn't get too much time to dive
That'd
deep
be
more.
cool. If it is convalescent serum, I mean, I'm always interested in in seeing stuff where they use people's serum.
So it looks. I mean, they compare about against like 10 monoclonal antibodies. Uh,
Okay, so maybe
uh,
it's there's a panel of antibodies to do it.
yeah, I feel like it is like a panel of antibodies that they use.
Yeah,
Um,
I'm not seeing the word serum appear,
yeah, I haven't seen that either. So I don't. Uh,
except bovine serum albumin, which is not the serum that I'm excited
yeah,
about. Okay, so yeah, that is very much like an extension of stuff that we were looking at last week.
mm,
Um, Very similar techniques, right? Like diving deeper into this idea of right where might the escape mutants appear, right? Because if we know they might appear in certain places, we also know what might be, like a beneficial titer. Maybe there's like key antibodies we could look for in people's titers, right? That's still an open question, I think.
Yeah. Uh,
Okay, next one. Oh, next one is this is like they published. I think they finally published like some. We looked at preprints when we were talking about the
right.
D614G thing, and so these guys they did some bioinformatics.
Yeah.
And they said that they actually they're trying to compare the hypothesis that we set out at the beginning to talk about as well. Is
Mm.
it a founder effect or is it a real biological difference?
Yeah.
And they use bioinformatics, and they actually find that they think it's a founder effect. But I actually find this evidence weak in the light of experimental evidence, right? Because if we're seeing isogenic changes
Mm.
make effects in animal models, if we believe that animal models are a good analog to the human infection, which again is also a little bit of a leap, and that's my bias, right? As someone who did animal models,
Mm.
right? I tend to think that animal models are a closer leap than seeing things apart. But yeah, so I sort of see that as maybe potentially weaker
Yeah.
evidence showing that
I
it's yeah.
because yeah, modeling it always depends on like what how you're analyzing your data, what data you get. So it can be, but I also see that there is a way for this to not be necessarily because it depends on how they measure in transmission. Because if we're talking about like the predominant like types, then then last week is basically looking at comparative transmission. So last time we looked at D41G versus the old version. So D61, sorry, new versus old, and in competition, the new one wins out. But that doesn't necessarily mean more transmissible, because with more transmissibility, you'd expect more cases or more average cases per
Absolutely.
genotype, and
Absolutely.
so it depends on how you're defining transmissible. Because if it is
Yeah.
like
I also see because it made me when I saw this paper, it made me rethink about the data that we saw in the preprint, and the transmission studies are competition first, then transmission in some ways, right?
yes,
Because they still right like because the animals are given the mixture, right?
yeah,
If the and we saw this too, like in the data, over time, the animals given the mixture, the D614G,
yeah,
um, that virus becomes more dominant in the animal, right? And then it is the one that is transmitted. So maybe transmission was not the right way to talk about this mutation, right? Like maybe there's a certain type of fitness advantage. It
yeah.
just doesn't sound good for the news. There's a certain type of fitness advantage conferred by this mutation, right? And that's led to its success, right? And it primarily through founder effects because of its intra host, right, in the
Hmm.
host competition with other viruses.
Yeah, so I mean, it'd be interesting to see like how they picked apart their models to try and come up with that. But yeah, it's
Yeah, so
uh,
like if we choose paper, it would be I do feel like it'd be very continuing that story, right, and trying to
mm,
mesh it with something that we already read.
yeah,
So actually,
uh,
these two papers, the first two papers of our potentials, like they're very much callbacks
yeah,
to stuff we talked about before.
yeah. Every YouTube
Uh,
channel needs its in jokes, and we've got ours ready.
just viral mutation and escape mutants.
Yeah, I admit we're a refined taste, but I mean, there's someone out there who'll be like, "Ha, D614G, I know what that means."
Okay, and the next one that we have on the list, I didn't I don't think this is a good one to discuss. It's very short. It
Oh
says
yeah,
consensus Spike CoV-2 potential implications in coagulation process and cell fusion. Basically, they did a bunch of sequence diving and they found a sequence that is found in like von Willebrand factor and other coagulation proteins.
yeah.
So
I mean, it's interesting. Because I mean this feels very much like a citizen science kind of like I download the data, I can analyze it and I can come up with sort of
yeah,
ideas.
I mean, that
And
would be amazing. Like I love that idea. Like, why
yeah,
didn't citizens find this? Why did we have to find some academic who's scraping the bottom of the barrel for stuff to publish, right?
because we don't teach people how to do this. We need to. We I mean
It's
that
true.
if
True.
we taught people how I mean Fold It was one of the great ways of like teaching people well getting people to do science and but the moment you have to like I mean I've read through these bioinformatic like kind of how-to guides and they are not user friendly. So
They, yeah.
they are because again
I,
they're designed
very friendly. I agree.
yeah they're basically designed for people who are reprogramming the entire software as they're using it. So because of that,
Mhm.
it ends up like being quite a bit hard for someone outside of that field to get into. But
Totally. But yeah, you heard it from us, citizen scientists. You could have written this paper yourself with knowledge of GenBank and knowledge of like protein annotation, right?
yep,
And knowledge of the literature. I think that's another thing. You can't. I don't want to be too disparaging on academics here, right? Because like that body of literature that they'd read to know to make this connection and then to
mm,
show us a bit of data, that is also a lot of work, right?
yeah,
Not every person is sitting in their homes and reading all these papers to be able to contextualize what they found in the body of literature, and that's
yeah.
still very it's a big part of the work here.
Yeah, I mean, it'd be great if they had like, was it Rosetta, and then they could model these interactions
Oh,
in real
sure. Yeah,
time.
like sort of give us like if they're a computational lab, then push it a little bit more, right? Show us like, right, some of those docking simulations, right,
Yeah,
of now this consensus sequence into the targets that you hypothesize are the ones that cause the issue. Yeah,
yeah,
absolutely.
yeah,
And,
Rosetta. That's another callback to a previous episode. In jokes.
science is just one huge in joke. If you,
Yeah,
if you,
basically, we constantly reference each other. We're like the Big Bang Theory in real life, where we're just going like, oh hey, remember last week when we looked at Rosetta data and didn't really understand how it worked, but we tried our best.
okay. Up next in the list, we have functional landscape of SARS-CoV-2 cellular restriction.
Yes.
I was
So
just diving for papers. This one I found, I think, on the Oxford preprint immunology. They talk about preprints and stuff.
yeah,
This one was there, and I think this is giving us host factors, right?
yes, they look at like all the various ways cells resist SARS-CoV-2, and like
And
kind of try and
because the title of this thumbnail has to be like callbacks to all the different episodes, but uh,
yes,
a lot of the ways that they find about resistance is the interferon stimulated genes. Uh,
yes,
it's a general antiviral response, and they're just going to go into detail showing that yeah, this is one of the ways that our cells use.
yeah. So I think
So
it's
it's a
good.
cell culture paper,
Yeah. So it's
a
very much
cell
like
signature cell culture paper. Sorry,
yeah,
I didn't mean.
yeah. So it really very much like goes into like the cell what happens within the cell. So in terms of like the whole system, it's a bit harder to, but it does a lot of like good molecular like stuff.
Yeah, what I like about this paper is they have this figure. I'm scrolling through, I'm seeing figure three, and they have like the life cycle of the virus, and then beside it, they have some um, experimental evidence of like the expression of certain host factors, right? And right, like, I think I like the point, and we haven't. I could talk about forever, right? But like infection is that interplay. We could talk about forever. The
Mm,
interplay between the host factors and the pathogen factors, right? And this paper is helping us understand those host factors a little bit better, right?
yeah. Hold on. Literally just like pulling up the figures because uh my PDF like takes a long time to load. This is a big paper. So
Yeah,
yeah they literally do have that. So that actually looks quite nice. That's quite interesting.
yeah. I just kind of like that like thought process, right? I mean, I'm not exactly sure what they're showing here because again, I'm just glancing at it and not reading the caption. But like, it's a cool figure. I'd like to talk about that one.
Yeah, I mean, yeah, it looks like the expression of certain like molecules at the infection stage based on their parental cells. So like the cells that they were derived from, they go like, oh, that's our control. Let's see like when SARS-CoV-2 binds, what happens, what changes,
Yeah,
and then
exactly.
yeah, uh,
Yeah, okay,
but
that's candid there. A long
yeah,
story, a long one. Next, we have association between ABO and Rh blood groups in SARS-CoV-2 infection or severe COVID uh, COVID-19 illness.
yeah, we haven't
So
done
this
yeah.
is this is a long line. We have not talked about this story, but
Mm.
there have been a lot of papers about blood type associations and infection. Um,
Yeah, I mean, this is I mean, we haven't really done any cohort studies because I do feel like we would end up dunking on them a lot because
yes, the
the
data is weaker, right?
yeah, the data is weaker, And it's when you're an experimental scientist, it's hard to like get that out of your head because you realize because it's more like a social science sort of thing. So you have to like take into those sorts of things into account and analyze it on its own terms. And
Yeah, well, I mean, it's sort of this thing like as experimental scientists, we'll never get like the human reality, right?
yeah,
Because we're working in models. But here, they have the human reality, but human reality is messy and complicated, and it has tons of these covariates, right?
yeah,
And a lot of the work is like statistically controlling and also through study design controlling those variants, also getting bigger sample sizes.
yeah. So it's very hard to do this study. So these kinds of work, and they are prone to so many like kind of confounders. So it's very hard.
Exactly,
And
exactly.
yeah,
And so the reason why I put this on the list today was just because we really haven't talked about this area, right,
mm,
of SARS-CoV-2 biology, where people have been trying to find these associations with almost everything,
yeah,
and blood type has been a particularly, I guess, rabid field of trying to find those associations. And I like the intro of this paper a little bit because it says very specifically the reason why they're trying to find this association is to direct screening efforts, right? So it's not
yeah,
about who's safe and who isn't safe, right? It's about if we could screen more people, are there certain groups of people that are easily testable for? We already have this information on
mm,
most people, right? That we should screen for more, and I like that, right? Like it also shows us that science is like it's as much about what we think that science is going to do for us as like what the reality it's showing.
yeah. Uh, so it's yeah. I mean, because
I
cohort
think it's
studies, yeah,
I'm gonna say I think it's probably not something we would cover because it also falls into that whole. It's a lot like a clinical trial where we're seeing a lot of graphs, we're not seeing a lot of mechanism. It's like a lot of stats. Maybe if we
yeah,
had some very enthusiastic stats person to join
yeah,
us, definitely consider doing more papers in this of this fashion.
yeah. I could. So if you in the audience, if one of you is a stats person or knows someone who is a stats person,
Yeah.
then
And
leave a
wants to
comment.
geek out with us on looking through every figure and answer our super naive questions about how things are done, yeah, we'd love to have you.
Yeah, we'd love to explore you. I mean, have you? Have you? Oh,
Okay, and the last one on this list we have recurrent SARS-CoV-2 RNA positivity after COVID-19, a systematic review and meta-analysis.
so yeah, this is something we haven't done systematic reviews and meta-analyses before. So
Once again, something that requires a lot of stats to fully wrap your head around. But we
yeah,
have talked about reinfection before.
yes,
Um, and so why I chose this paper again is because I thought it's interesting. Like we've heard all these case studies about reinfection that it happens, right? And they prove through genetics that like, oh wow, it really was a separate viral uh, isolate that
yeah,
infected these people. But what do we know about it, like in the general? Like how to zoom out? Tell me how common is it for people to
yeah.
get SARS-CoV-2 after getting it before?
So the thing about this is we've seen a lot of case studies on that kind of positivity after infection, but we haven't seen anything pulling those sorts of things together. And this is an opportunity to look at that, to examine that question, because case studies are very much you can't really analyze them. They're very much a situation. The only analysis you can go up to them and say, "Oh, you're a liar" or something like that. It's just there's not really much analysis we can do, but with a systematic review and method, we can do a bit more.
I'm hearing this. Hmm.
Yeah.
Hmm. Absolutely. Yeah, sorry, I heard everything you said just like in choppy
Oh yeah,
bits.
sorry. Yeah.
Are we are we
We're
back?
back. We're back.
I agree with what you said. Yeah, this is what is holding. That's why it's an interesting one to talk about. But I'm not as enthusiastic about it as maybe others.
Yeah.
Um, and I guess the only thing that I want to throw back into the mix here, then, so like, all right, we remember we have our Zotero that people can follow along, the long list of papers that we have put up for potential analysis. We try to sort them into places, and then also you can see some of the background papers that we use to then discuss the main paper. Link in the description
Yep.
below to the Zotero
Yep.
group. Okay.
Link. Oh.
Uh,
Yep. Everything
and so Yeah, and so the other thing that I want to throw out there is there was the back-to-back papers in Nature about the other receptor, Neuropilin.
Yes. Yes. We can take a look at those. Those are quite interesting.
Yeah, that might, that's sort of some interesting mechanistic implications when it goes to the lack of smell and some of the neurodegenerative aspects that people symptoms that people have been reporting. And then and so yeah, what were those ones
So
called?
Neuropilin one facility.
I'm in the potentials folder of the. It's under selection and then potentials.
Yeah.
Um,
So,
and there are two papers. One's Daly et al. And the other is Cantucci. Neuropilin 1 is a host factor, and neuropilin 1 facilitates. And then the other one I want to throw back in the mix is maybe pro-thrombotic autoantibodies in serum from patients. Zhu et al.
okay. Let's see if I can pull up that quickly. Yeah. So okay. Right. Yeah.
Yeah, because I think that there's a lot in the back
Yeah. We've
catalogs
got
of
a
our stuff.
lot. We've looked at a lot of papers very briefly, but there are a lot of like good ones in here. Um, so I mean, I'm kind of partial to the Neuropilin one paper. Uh,
Neuropilin 1.
so
Yeah, Neuropilin 1.
can
I like I like the functional landscape of SARS-CoV-2 cellular restriction, although that is maybe a monster of a
yeah,
paper.
we have given ourselves some monsters of a
Uh,
paper recently. So
yeah, I'm not really into the callbacks, just because we have done
yeah,
them
no, that's right.
before.
Uh,
Consensus, we're not going to. Right, so we have this functional landscape, which is a big one, and then we have prothrombotic or neuropilin. So let's see how big these neuropilin papers are, because they were published back to back in Science.
yeah,
Um, so one is quite short, maybe 3 figures. Pretty good. Actually, they're 3 big figures plus the supplement. That's the Daly et al. Neuropilin is a host factor.
right. Okay, I'll pull that up. So
And then the other one is a 3 figure paper. Also some bigger figures
okay.
with S10. Oh my god, with tons of supplements. It goes up to S10.
I mean, last week we had S20. We had 20 supplements, so I feel like we're strong
Yeah.
enough now.
Oh yeah, we could definitely do it. No, I guess what I'm saying is, that one might have more material we can look
Yeah.
at, because it looks like the daily one only has um, 5 supplementary figures. I don't know. They both kind
I
of have
mean,
a similar amount.
I'm kind of like interested in the like Cantucci Castelvetri paper. Um,
Okay, cell entry and infectivity.
yeah, mostly because the title catches my attention a lot more. Because I
Sure.
think I got clickbaited into it.
Oh right, because cell entry is more declarative than host factor.
Yes, that is basically it. So, yeah, yeah, that's right. Yeah, yeah. Okay, there are links for everything we've talked. So I know there are misinformation. So I'm trying to illustrate by linking to everything, even though our comments are really long. But yeah, it's
Absolutely. And if you do find mistakes in what we say, right, like please let us know. We are interested in being a reputable source of information, and we only know what we know. We try our best to say things, but sometimes in the heat of the moment, stuff might come out. And yeah, we need our audience as much as
well,
you
yeah,
might need
it's
us.
a peer review process. We're peer reviewing, you're peer reviewing us, and we're all kind of it's like a surveillance state, but everyone's like Big Brother. We're all brothers. We're all family.
I was like the needle of like scariness, like was like, oh my god, what's he saying? Oh, okay, I
Yeah,
guess we're family.
this
It's
exact comment is an example of why we need people to tell us to shut up sometimes. Or tell me to shut up. No, Danny has done nothing wrong.
Oh, please don't say that because that's not true. All right. It was
Yeah,
great to talk with you. It was great to catch up and hear about the headlines. For the viewers out there, if you want to follow along, please read the paper with us. Tweet us your questions. We'll do our best to answer them. And join us next week where we'll dive in figure by
yeah.
figure.
So see you next week.